So we've now just understood how triglycerides are formed in our bodies. And the next thing we want to talk about is a phospholipid bilayer. And hopefully you've watched the lipid videos where you understand the rules of lipids in our bodies. So what is the phospholipid bilayer? You're probably hearing this in your biology class and something with cell membranes, and you're like, what the hell is this all about, man? I don't get it. Well, if you're thinking that, by the end of this video, you're not going to have that question mark in your head. So a phospholipid bilayer is basically another form of triglyceride. That's not exactly true because I'm going to get that to in a second. But what the essence of the phospholipid bilayer are also these fatty acid chains. So we have a hydrophilic and a hydrophobic part of the phospholipid bilayer. And imagine this is the membrane of a cell. That's a bad drawing right there, but hopefully you get the idea. The whole cell is behind this. We have the cytoplasm right over here, and that's the that's the outer side of the cell. So what's happening essentially is this phospholipid bilayer gives a very unique characteristic to the cell. So we know that cells are floating in a solvent, which is water, and therefore it likes water. Whatever likes and attracts and is attracted to water, meaning it's polar, goes on the outside, and that's the hydrophilic part. And whatever is not liked by water and repulsed from water is the hydrophobic tails. And we've already discussed what hydrophobic means and hydrophilic means. So what is the phospholipid bilayer made up of? So we recently discussed what a glycerol molecule was and its role in the triglyceride molecule. What really happens and how these phospholipid bilayers are made is essentially very similar to triglycerides. However, notice that this is a triglyceride molecule right over here. So a triglyceride molecule has 3 different fatty acids attached to the main body of the glycerol molecule. This is the glycerol molecule right here, and this is the ester version of the fatty acid that is attached. Notice we have 3 different fatty acids, but when we look at phospholipid bilayers, it's very similar in the sense that we have two different carboxylic acids, fatty acid chains. This is bothering me. Why is this not being focused? Excuse me. Give it a second. Anyhow, so we have two different fatty acids that are, thank you, two different fatty acids on this chain, on the binding to the glycerol molecule, and then we have a phosphate group. That's the only difference from the triglyceride molecule. And what really happens is this is what it looks like. This is the base structure of a phospholipid bilayer. This is called a phosphoglyceride, and we call it a phosphoglyceride because we have a phosphate group and then we have a glyceride, and that should make sense by now. So we want to really quickly talk about how saturated and unsaturated fatty acids affect the phospholipid bilayer. Now, really quickly, saturated fatty acids are all single carbon-hydrogen bonds, and unsaturated fatty acids have at least one double bond between the carbon and the carbon. So now I want to ask you this question real fast. If you watch that video where I talk about unsaturated and saturated fatty acids, you know that if we have a unsaturated fatty acid, then there is a little bent in the entire structure of the fatty acid, and that kind of does something in reference to the membrane fluidity. So here's a question I want to ask you. Would you think that saturated or unsaturated fatty acid increases membrane fluidity? Now, I've given you 2 minutes to think about that. Not really, but still. So kink shape, the bent structure that's formed by the unsaturated fatty acids will reduce, will give its kind of like a bent shape, and we've already talked about that. It's going to reduce the van der Waals forces between the long carbon chain, and what that means is that there are less interactions and there's a less binding between the entire chain. That means there's more fluidity, and that's why we have double bonds, or unsaturated fatty acids, increasing membrane fluidity. And this is going to make more sense as we proceed and understand the membrane structure. So another question I have is, do you think that a longer or a shorter chain, do you think that a longer or a shorter chain will have a higher fluidity? Of course, it's written over here. Longer chains will have more interaction because think about it. We have a higher number of carbons inside the structure. We have more carbons, so there's a higher sharing of electrons between everything, and hence there's more van der Waals forces between everything. So the shorter the carboxyl, the shorter the fatty acid, the more the more fluidity in the membrane. So here's an extra extra information that we will talk about later on. So cholesterol. We have probably talked about this in biology, that cholesterol is a part of the phospholipid bilayer. And what cholesterol does is at optimal temperature, it increases membrane fluidity. And here's a very quick sketch of the phospholipid bilayer. So in the phospholipid bilayer, we have we have the hydrophilic stuff on the outside, water-loving stuff, and we have the hydrophobic stuff on the inside. On the inside, we also have, this is the membrane, by the way. Whoops, let me just use a different color. This right here is the membrane. This is just a bad representation, but get the point. And then inside this, we have cholesterol. Cholesterol is something that's regarded as really bad in our blood, and we're going to understand that more. But inside the cell, inside the phospholipid bilayer, it's a good thing. It's increasing membrane fluidity. But at high temperatures, these cholesterol molecules can reduce fluidity. Now think in terms of kinetics, and think that this kinetics is high. Everything is moving really fast inside the phospholipid bilayer, and because of this fast kinetics, the fluidity is decreased as temperature is increased. That's a very quick rundown of phospholipid bilayer.